Today, we would like to invite Dr. Ariane Helmig to give this talk under the organization of InterPore Academy lecture. So I would like to spend a few minutes to introduce Rainer. Professor and Dr. Rainer Helmig was born in Germany, and in I suddenly found it's almost your birthday, right? Happy birthday!
That's true. That's true. I agree with you. Not so far.
Yeah, and Rainer got his PhD in Germany, and he's now a full professor for hydromechanics and modeling of hydro system. He's now a director of the Institute for Modeling Hydraulic and the Environmental System at University of Stuttgart. Actually, Rainer has got a lot of honors, including the Honor Lifetime Membership Award from InterPore. And last year, he was elected the AGU Fellow, a very great honor for the geophysical science. And Rainer has published a lot of papers and have done a lot of work on the multiphase transport in porous media with applications in energy, in life, and bio and medical science. So I would like to leave the later time to Rainer for the wonderful talk. Thank you.
Thank you very much for this very kind and friendly invitation. And it's my pleasure and honor to present you a little bit more stuff about our multiphase flow and transport research related to complex porous media. And the title for today is...
Rainer, can I add one sentence, please? Yeah, actually, Rainer has gave this talk 20 days ago, and that time is almost middle night in China or Asian time. It's very hard, it's very tough, and the InterPore Academy arranged another chance for the Asian audience to listen to this wonderful talk. So that's, yeah, I have to add. Yeah.
Thank you.
Yeah, please.
Thank you.
Sorry.
Thank you very much. No, it's okay. And it's really my pleasure to welcome and to give this talk to the Middle East friends from InterPore, from Australia and China and Japan and so on. And again, the title of my talk is for today, From Brain to the Fuel Cell, Porous Media Are Everywhere. And you can trust me, I try to do my best to show you that porous media is everywhere, okay? And these are the classical fields of application. You see here, for instance, this is a test site that we have analyzed with colleagues from Harold Watt and Stanford together. That was a CO2 injection parallel to this geothermal, the optimization of geothermal energy. Then, for instance, this is the Johansson formation. We have analyzed this for CO2 storaging. And last not least, this shows here fracture matrix interactions plus dilatation. That means the opening and closing of fractures. And this was developed especially for atomic waste disposal sites. But porous media is not only related to the geo part, it is extremely relevant for other fields too. And I will explain you a little bit more why and what kind of tools do we need, what kind of techniques do we need to analyze very complex flow and transport processes in bio, in technical and environmental systems. That's the team, that's our team from our department. You know, we have a new member, it's Majid. Majid is now a senior professor in our team, and this is our whole team, postdocs, PhD students, and 3 of us are professors, Holger, Bernd, and myself. Okay. From the brain to the fuel cell, and I would start with the second part of my talk. Porous media are everywhere. You know that it's a piece of concrete, and you can see 2 colors in it. This is the yellow and the blue one. It can be that there are 2 fluid phases in it. The yellow can be the gaseous phase, and the blue one, the liquid one. And is this relevant for other fields of application? Yes, of course. I will show you a few of them. That's our skin. Our skin is a very complex porous media system with 3 different kinds of layers. And the climate of our body is strongly dependent on our skin. That means, for instance, if we have hot days, then we have this evaporation process takes place. And if you have this evaporation, then you get a cooling of your skin, of your whole body. That means it's a complex porous media. It's a bio one. The next one is, this is the bone. The bone is a very complex porous media. For instance, if you have glass bones, then the question is how you can stabilize this, how you can inject different kind of gels into the porous media, into the bone to stabilize this. The next one is the brain. I will come back to this later. The brain is an extremely complex porous media system. Then an onion is a porous media. We haven't done any kind of research related to an onion, but onion has a very complex porous media structure with different layers. Then a leaf. A leaf is a very complex, again, porous media system. Why? Very simple. The evapotranspiration and the whole interaction between the soil and the plant is organized by the leaf. And the thickness of the leaf, the structure of the leaf is related to a very complex porous media. And inside there is really a multiphase flow system that controls the whole interaction. Then, of course, our unsaturated zone. The unsaturated zone is really the interface between the atmosphere and the water cycle. And on one hand, on the other hand, we need our unsaturated zone, especially for our plants. That means, for instance, that is really a highly complex porous media system. Then, of course, if you have a top view on this and you have related to this climate change, for instance, higher evaporation rates, then it can be that you get a change of the structure. That means, for instance, you get shrinking. And if you have this shrinking here, then of course you get fractures in a system. Last not least, a technical application, I will come back to this later on, is a fuel cell. A fuel cell based on 90% of porous media. And this porous media controls really the interaction of hydrogen and oxygen to produce electricity. I will come back to this later on. It's a highly complex porous media system. Let me start at first with the bio part. And I will explain you, or not, sorry, I will explain you what I have in mind. You see here, that is an abstraction of the porous media on the pore scale. The yellow one is one fluid phase, the blue one is the other fluid phase, and the gray one is here the solid part, the grains. That's the porous media. The porous media can be interact with, for instance, a free flow system, like a tube or the atmosphere and so on. And it can be that if it's a fracture or a tube, that on the other side, a porous media system is again. The question is now, what kind of models do we need? How is this necessary and important for different kind of applications? Okay. The first one that I would like to discuss with you is related to a biological system. For instance, this is the brain here, and this is a part from a brain. That means, for instance, you have the vascular system. The vascular system has 3 different parts: this capillaries, tubes, the Mullins tubes, and the arterial tubes. And this is embedded in the tissue. That means, for instance, these are the vascular system here, and this part describes the tissue. The tissue based on 2 parts, the white and the gray matter. And why is this so important? If you think about therapy, therapies about tumor therapy or multiple sclerosis, you need a clear understanding about this kind of interactions. I will come back to this later on. The second one is more related to technology. I mentioned this. This is a stack of a fuel cell. You have now here the gas distributor that's related to this part here, where you inject hydrogen and oxidant. And then, of course, related to these diffusive layers, that is like this porous media here, this green, red, and the yellow one, we get this mixing reaction. And based on this electrochemical process, you can produce electricity. The last example that I would like to show you is in the same range, based on the same model concepts, is related to the environment. That's related to root uptake. You see here, this is the unsaturated zone. This is a plant here, and we are interested, how does the root interact with the solid? That means, again, the unsaturated zone, the solid part, is related to the porous media, and the root is more related to this green free flow part. I will explain this later on. Let me start at first with the concept. The concept is now how we can embed complex tubes, 1D, into a very complex three-dimensional porous media environment. Porous media plus transport network. That is the key topic for today. Let me start at first with the brain. I mentioned this. So that is based on the PhD thesis, a doctoral thesis from Timo Koch. He's now a postdoc in Oslo. The brain, a porous media with a complex structure. We have different brain cells, blood vessel network, and cell interstices. That means, for instance, we have structures of 100 nanometer. Interstitial fluid. What does it mean? That means, for instance, we have the tissue, and that is the three-dimensional porous media. And then, of course, we have this pipe system, that it's the vascular system. You see it here. And we will analyze later on the multiple sclerosis and how we can use this kind of model concepts for the better analysis of multiple sclerosis. This is a picture that we got from our colleague and friends from the University of Utrecht, Franca and Bruno. And all this kind of work is done in cooperation with the University of Bern and the University of Zurich and the ETH Zurich. Let me come to the details. What's going on? At first, we must identify the structure. Where are the vascular system? Where is the tissue? And how they interact with each other. That means we need an imaging technique. And one of them is MRT. That means imaging process is an imaging process in medicine. Use differences in of different porous materials. You can identify bones, soft tissues, water retention in the porous space, and so on, to generate imaged contrast. And this, for instance, is now the patient here. This is the brain of the patient. And this describes here the vascular system, okay? And we get a strong magnetic field, non-harmful radiation like X-ray. It's important to know. So now we extend this, we do a perfusion MRI. That means time-resolved image series. And I will show you a time-resolved image series of about 2 minutes. That means what we have done, the colleagues from Bern in this case, we have inject a tracer, a radioactive tracer in this case, and then we can detect what's going on in the brain with this tracer based on the transport mechanism inside the vascular system and the surrounding tissue. So, and examples of use is examination after stroke, for instance, or examination of brain tumor, I mentioned this, or examination of multiple sclerosis, and I will come back to this later on. Knowledge of the transport processes and the structure of the porous media enables the determination of important physiological parameter. And that can be blood flow, blood volume, it's very important. And is the contrast media leaking from the blood stream? That's very important for multiple sclerosis. You know, in the brain, you have the brain-blood barrier. That means if the brain-blood barrier is in a good shape, then there isn't any kind of transport from the vascular system in the tissue over the interstitium. But if this is not the case, if you have this leakage, then we must take care. Why? Very simple. Then you are under not good conditions. Let me say it like this one. So what's going on? Similar. What is the reason for this? For us as modelers, as porous media researchers, simulation models help to analyze the image data more precisely. That's the idea. So let me come now to the example. That's a perfusion MRI. And for instance, this is one slice. And you see here the whole brain image. We have imaged the whole brain with 19 slices, 80 times over 2 minutes. This is one of them. That shows here the density differences with different colors. Slice image results resolution is this one, 256 to 256. So now you see here, this is the analysis of the system over the 2 minutes, and you see the different colors here. I will do it again. You see here that we get really, based on the density of the system, a very nice understanding of the system itself. Now we inject here the tracer into the vascular system, and we have analyzed 2 points in the brain. One here where we have a reference one. That means where the system itself is in a under healthy condition, and where on the other one, where we have an inflation. That means, for instance, where the blood barrier is damaged. Okay, if the blood barrier is damaged, you can see it with the whiter part here, spot here. Then, of course, we get a completely other breakthrough curve. You see here the 2 breakthrough curves for the reference one and for the inflation one. And there are differences. I will come back to this later on and how we can use this for the analysis of multiple sclerosis. Okay, so I mentioned this. Curves differ because contrast media leaks from the bloodstream in the inflammation tissue. In healthy patients, the contrast media does not leave the bloodstream in the brain. That means that's the reason why you get differences. Why you can identify, is this a risky situation or a not risky situation? But the question is now how we can quantify this. Is this really risky for the patient? With this modeling now, we can do the following things. How much comes out? We can quantify this. How much are the blood vessels changed? How can the analysis be transferred to other parts of the brain? Then double slope, double contrast media leakage, linear or nonlinear connection. That means, for instance, how intensive is it? Can we quantify this? These are the key questions if you are interested to analyze this and to model this. Okay, scales is a very important issue. And for instance, this is, for instance, my brain with a lot of holes in it. The brain is normally in centimeters, and the MRI voxel scale is in millimeters. And this, for instance, here describes MRI voxel system. I will come back to this. So this, for instance, is a single MRI voxel. This is from the red here. We have done it in cooperation, I mentioned this, with Patrick Jenny from the ETH Zurich and Johannes Reicholt and Bruno Weber from the University of Zurich. I was involved in this PhD activities, and we have analyzed here the different parts of the vascular system interacting with the porous media. You see here, we inject now here the contrast agent. This is and now our interest is if there is a brain-blood barrier problem, how much of the contrast will be migrates from the vascular system into the surrounding tissue. Okay, so you see here the dimensions. That's 1 millimeter in this direction and 2 millimeters in this direction and 1 millimeter in this direction. Okay, you see here now what I've mentioned so far before, the contrast agent. And this is now the red point here that I've mentioned so far before, where you have this inflation. And now you get this migration of the contrast agent into the tissue, and you can quantify this. You see it very well here. That's a simulation that we have done for the red, and we have used these models. I will show you this for real patients too. Okay, I hope the philosophy and the strategy is a little bit, it's clear now. What kind of mathematical model do we need? What kind of model strategy do we need? How can we describe flow and transport process in the porous media with embedded tubular network systems mathematically? That is one of the key problems that we have. Challenges. Okay. And again, example here, tissue perfusion. So this is, for instance, a slide of a very complex porous media. That means, for instance, the red part, the darker red part describes the vascular system, the whiter one, the tissue. Okay. We have done an abstraction. I mentioned this. We have approximated on one hand the vascular system, and then we have embedded the vascular system in a surrounding tissue. Okay. So we have two systems now here. We have on one hand the tissue, and we use the different kind of homogenization techniques to get this cell structure into a porous media structure, where we can use volume-based quantities. On the other hand, we have a second system that is the vascular one, and the vascular one is then directly embedded in this tissue system that is averaged before. This is one of the publications related to this homogenization that we have used here. Okay. The next one is how we can approximate the vascular system. It's a highly complex system. If we go into the details, then we have on one hand here the cross section. This is the endothel. That is a collection of cells that builds the barrier, the wall between the tissue and the surrounding, the vascular system and the surrounding tissue. What we have done, we used a special kind of homogenization to get this wall. Then we have transferred this thickness of the wall into a special diffusion function. And then we have reduced this three-dimensional system into the 1D system. You cannot calculate for this kind of complexity 3D flow and transport field. What does it mean? That means, for instance, here we have a Navier-Stokes equation. We have particle dynamics and fluid structure interactions. And now we have transferred this to a Persoy type flow, very simple one, plus effective viscosity and effective description of the wall itself with a diffusion coefficient. Okay, that's the first simplification. The next simplification, oops, that was too fast, is how we can embed this into the 3D system. That means equation governing fluid flow, coupled Persoy-Darcy type equation. We used for the vascular system, I mentioned this, the Persoy equation, and for the surrounding tissue, the porous media, the Darcy equation. These are the equations that we have used. I'm 100% sure familiar with the Darcy one. Then with the continuity equation, and this describes the exchange. And we have two exchange fluxes. One is based on the pressure difference, the classical viscous flux, and the other one is related to the differences in related to the behavior of the wall itself, that is like a membrane, and then we can describe the pressure related to this membrane. But the question is now, what's going on with and this equation is so-called the Starling's law. And what's going on now with this exchange fluxes, the exchange between the vascular system and the surrounding tissue. That means the Q and the phi here. And we have spent a lot of energy related to this to get this embedded as good as possible into the 3D arbitrary environment. So you see here the gray part describes the porous domain. This is the tube here with the dashed line. Then this red one describes the source distribution, and the blue one describes the evaluation and the reconstruction. What does it mean? That means the red one here, type of mass distribution in the porous domain, line, surface, volume. That means that is the influence of the source from the vascular system to the tissue. The blue one, this one here, types of evaluation and or reconstruction operator needs to evaluate for the source to get this source into the porous media. These are the two parameters that I've mentioned so far, as describes the exchange now between the tissue and the vascular system. So now let us solve this as good as possible. That means what we are doing here, it's a long straight vessel embedded in the tissue. And I mentioned this, we can calculate with this strategy now independently in one complex environment, the tissue and the vascular system. That means this one is embedded in this one. Okay. So what we have done here, we have developed, there are different kinds of papers. One is accepted this year, where we have analyzed this and we have developed then a distributed source method. We call this DS method. That's done by Martin and by Timo. And Patrick Jenny was involved in this too. That means we have now analyzed this maximum pressure. And then, of course, we have transferred this maximum pressure of the 1D into the 3D environment. That was the idea. If you're interested on details, please let me know. Okay. So that's the blue one here. And then, okay, let me come now to the application. Uncovering subvascular processes using computer models. The question is now we have the tools in our hand. We have the model in our hand, how we can handle this, how we can generate really help for the medicine, for the patient. And at first, we used the numerical model, a forward model, okay? And we analyzed really the concentration evaluation in the vascular and the extravascular space. That means that we get an understanding on one part, tiny part, how it works, how these two parts interact with each other. In the next step, we approximate the MRI model. That means, for instance, we include really the information that we have from the measurements. Then, that's published here. Then we use the volume averaging over this. That means, for instance, that we have now one voxel analyzed, that we have one voxel now transferred in a mean averaged concept, so that we can use this as the basis for the whole. That's the idea. Okay, to get really the breakthrough curves that I have presented you before. So what do the simulations show? Example, determination of the effective diffusion coefficient of the contrast media through the capillary wall. How strongly is capillary wall affected? It means how is this damaged? Okay. The next one is simulation-based MRI pixel analysis by inverse modeling, so that we get really a good field of parameters. The next one is model-based quantitative parameters estimation and quantification of parameter uncertainty. How uncertain these different parameters? Which one is more important in comparison to the other one? And the goal is better basis for the decision-making by treating physicians. That means to support the physicians and to give the patient more help. Okay, patients more help. And you see on what's going on. This is, for instance, for the problem. That means for when you have this problem with the brain-blood barrier, and this is for a healthy patient or healthy part part of the brain. Okay. And now we have analyzed this, and I will show you this, how it works. This, for instance, is the selection sensitive model parameters. That means we have inflow profile, we have MRI signal, we have transmural exchange coefficients. And this is the breakthrough curve. So, and now we have done a parameter optimization. We used here a maximum likelihood estimation. And these are measurements. You see here the measurements for the inflation point. And these are the measurement for the healthy one, for the healthy data. I call this healthy data. And with this technique, we can approximate now very well the measurements. And now we can use this model for the analysis of different patients. That was the strategy that we have developed. And this strategy is now used from our friends in Bern and Zurich. I hope this is clear. So now you have got an impression, only an impression, how tricky it is and how much fun it is to combine 1D with 3D for biological systems. The question is now, can we use this kind of equipment, this kind of knowledge for other applications? Yes, we can. For the environment, that is done by Katharina and Timo in cooperation with colleagues from Belgium and from the Environment Center in Jülich, Jan van der Borg. And you see here, this is related to root uptake. I mentioned this before. That's a root-soil interaction and root growth. That means, for instance, this is the porous media here. And now the question is here, is this 1D root, how we can embed this 1D root in the 3D environment, in the soil, and how we can handle really mass momentum and energy transfer, and of course, the growth of the root. These are the key questions. Let me come to the system itself. Vedozone processes. On one hand, that is the plan here. We have this porous media, the unsaturated zone. Then, of course, we have here the flow and transport in the root. Then we have the porous media. It's a multiphase flow system, 3 phases, solid, gases, and the liquid phase. Then we have this root uptake, where the root grows. Then we have the root tip. The root tip has a special structure. And of course, we have evaporation and evapotranspiration processes. And of course, the soil, the surface of the soil interact with the atmosphere. We have soil evaporation. That means a highly complex interaction of different processes. So we have analyzed this. I will show you, it's only a showcase, nothing else. This is really the root-soil interaction. These are simulations that we have done. And you see here, this is the root system. And the different colors shows the flux in kilogram per second. That means you have a high flux on the tips of the root. And if the roots are bigger, you get less. It's clear, continuity equation. And we have analyzed this for 2 different cases, for a sunny day and night. That means on one hand, we have an intensive evapotranspiration and evaporation process. On the other one, we have less. Okay. Can we simulate this? Can we analyze this with the tools that I've presented you before? And this, for instance, this was for the sunny day, and these are the results for the night. And you see the intensity decreases. Of course, you have less evapotranspiration processes, and of course, that means that the intensity is less. You can see it here. We have analyzed this for a soil evaporation and plant transpiration, evapotranspiration rate in millimeter per days. And you see here the total one, that's the black solid one, then the evaporation process, and then when we have included transpiration, and you see the differences here. Okay, you see we can analyze the influence of the different weather related to the growth, the root uptake. And that is the, then of course we have analyzed different kind of systems with and without root to see really how big is the difference. And you see, if you have plants in it, then you get the overall the black one. And if you have only evaporation, then you get the purple one. Okay. But the key question is now, can we simulate really the root uptake? And this is in cooperation with Andreas Schnepp from the University of Bonn in Vienna, where we have used the model that I've presented you before. The only extension is that we have developed on the tip of the root a special algorithm that describes the growing of the root. The growth algorithm adapted from this toolbox based on the work that from Andrea Schnepp's group. And we have compared this with real measurements, and that looks not so bad. That means this is this one here, sorry, that's this one here. These are measurements here, and this is the simulation. And if you like it, you can have a look to this paper that I've mentioned. You see that the comparison study that we have done was not so bad. Okay, you see, you can use now this kind of techniques, this kind of model concepts in cooperation, of course, with other ones for different kind of applications. Let me use now the last 10 minutes or 5 to 10 minutes for another field, more related to technology. That is related to the fuel cell. And this part of the presentation is based on different activities. This is Katharina Barber. She has developed the first model related to this. She is now one of the leading persons for the development of fuel cells for BMW in Munich. Then Cynthia has finished her PhD last year. She has worked for aerospace, better aerospace concepts for drop porous media interaction. Cynthia is a PhD student from Bosch, where we work together, especially on fuel cells, and that is Maziar. Maziar is working on the drop formation and the better description of drop interactions between free and porous media flow, especially under hydrophobic conditions of the material. I will come to this. So let me start. Why is this so important? Very simple. Fuel cells, don't they already work? Yes. A fuel cell is a factory that transfers chemical energy to electrical energy. But efficient water management is necessary. I will show you why. And the motivation for us is the following: enhanced process understanding, identification of relevant processes and parameters, and the optimization of gas-water budget in the fuel cell. Maximum power output, that is the key aim here. And for instance, this is a Mercedes bus. You know, the headquarters of Mercedes is here in Stuttgart, where they analyze this for buses and for bigger tracks, and of course for other purposes too. So let me come to the details. That is a polymer electrolyte membrane, the PEM fuel cell. Chemical reaction energy of continuously supplies fluid and oxidant is converged into electrical energy. I mentioned this. The chemical energy is converged directly into electrical energy. That means, for instance, you inject on one hand the H2, on the other one O2, then both of them will be mixed with each other in the so-called reaction layer. Related to this reaction, this electrochemical process, you produce on one hand electricity and on the other hand water. And the problem is now the water must be extracted in the same area where you inject the O2. That means, for instance, if you need a lot of energy, lot of electricity, then you produce a lot of water, and it can be this water blocks then the flow of the oxidant to the reaction layer. That's the key problem. That is the reason why we call this we need a good water management. This is the stack that I have presented you before. That means, for instance, this has different layers, and I will come back to this in a minute. So, and this, for instance, is the gas distributor here, where we inject on one hand the H2 or the O2. Okay. So this is the stack that I've presented you before. This is the end plate with integrated flow field. This is the gas distributor. Then we have the anode. It's a diffusive layer. It's a porous media. Then we have the membrane, the red one. That's the porous media. And we have the cathode. It's a diffusive layer. It's a porous media too. What does it mean? This describes really the interaction with free flow and porous media flow. You have free flow here in the gas distributor, and then we have a multiphase flow behavior in the porous media, anode, membrane, and cathode. Okay, how we can handle this? This is the real picture of this kind of system. Okay. At first, we must think about the scales. Their scales are very important for us. This is the typical length scale here, and that's the typical pore radius. And we are really interested on this one here, on this area here. The question is now, what kind of techniques, what kind of model concepts do we need to approximate really the relevant processes, the multiphase flow and energy and phase transfer mechanism and processes inside the diffusive layer? Okay, let me come to the details. Again, fuel cell, how do the porous layer interact? That's the gas distributor here. That's the diffusive layer. That's the reaction layer. I hope you can remember this. If you go a little bit more into the detail, what kind of model concepts do we need? On one hand, we have here the free flow system. Then, of course, we have the interaction with free gas, drop formation and separation. Why do we get these drops? The diffusive layer based on hydrophobic material, Teflon, like Teflon material. That means, for instance, the gaseous phase is the liquid phase, and the gaseous phase is the wetting phase, and the liquid phase is the non-wetting phase. Very important to know. And then, of course, we have here the reaction where we produce the water related to this electrochemical process. Okay, so that means, for instance, we must have an understanding of the multiphase flow processes inside the porous media. We must have clear understanding of the free flow regime and, of course, the growing of the drops and how they interact with the free flow. Okay, that's the key problem here. That's the reason why I've separated this in 3 layers: the free flow part, the porous media one, and the part where the drops grows and interact with both the free flow and, of course, related to this growing process to the porous media. That's the idea. So, and what kind of processes are relevant? On one hand, the growth and the formation of the drop. If possible, the merging of the drops and the detachment. If the drop is big enough, related to the forces from the free flow, you get this kind of detachment. What we have done, we have integrated over this, I will show you this in a minute, to get a new interface. That means we have now a very complex interface where this kind of processes directly included. And we have developed for this a multi-scale approach where we combine the free flow with the porous media flow. And the interface here is described and approximated with a multi-scale approach where we locally approximate these processes and globally integrate this into the combination between the free and the porous media flow. The only equations, it's only to give you an impression where we are. Okay, for the free flow, we have we use the fully Navier-Stokes equation with the inertial term. Then we can describe the total mass. We have the component of each system. That means it's water, air, and if you have other H2 and so on. And then you have the energy balance equation with the internal energy, the enthalpy, and the temperature. And these are, for the experts, these are the primary variables. You need the variable switch. Why? You can have phase change processes inside the porous media and at the interface between the porous and the free flow one. Okay. The porous media, we used very simple classical multiphase flow concept with the extended Darcy's law, total mass balance, component mass balance for each components, and the energy equation that I have presented you before. The only term, the only part differs to the energy equation before is the storage of energy in the solid phase. Okay, the same. And these are the primary variables, and we use the different kind of switching techniques to switch from single to multi to describe really this phase change. Okay, now we come to the tricky part, the interface. Here, this kind of interface here, I mentioned this. We must have water drop on soil surface, here, water drop on a pore throat. Then we must think about surface tension, contact angle, and hydrophobicity. That means, for instance, how does this interact with each other? Then we must have a clear understanding of the forces in the surrounding of the drop. When does the drop detached? That means we have drag forces on one hand and retention forces. That describes really the force balance in the surrounding of the drop related to the free and the porous media one. So we have done it like this one. We have the formation. That means based on the capillary force. Then the growth due to liquid flux from the porous media and shrinking. If we have evaporation, then of course we get now an evaporation process from the drop to the free flow. That means if you have a very intensive evaporation process, big temperature differences, then of course you get a shrinking of the drop related to this kind of processes. These processes interact with each other. Then of course you can have deformation due to drag force. And of course, I mentioned this, you have this detachment. That means, for instance, the question is really, one is the drag force bigger than the retention force? Okay. Then of course it can be if the pores are interact with each other, then we get the merging, that one drop merged to another one. And then of course it can be that we get a film here on the surface between the pores and the free flow one. These are the different aspects that we must include in this very complex multi-phase flow system. Last common numerical methods, if you are interested on this, we used a very simple staggered grid for the free flow and a cell-centered one for this case for the porous media one. We have a conforming grid, and we used all this kind of things monolithically in one system. Okay, and we used for the time discretization implicit Euler scheme. So to make a proof of all these kind of things, we have done it in cooperation with the University of Bergamo from Italy together. Stefanie Santini, she has done measurements. That means what she has done, she has prepared an experiment. Pore is Teflon, hydrophobic, block connected to a pump. Simulation on the drop, that means, for instance, they can measure now. Here, this is the pore, the flow inside the pore, the growth of the drop, and if necessary, the detachment. And you see here the results of the growth of the drop on the top view. And this is the comparison study that we have done with these measurements from the colleagues from Bergamo on our simulation. And that looks not so bad. It's not published, but I hope we can publish this soon. So let me summarize this now. And now the question is how we get really a model that is relevant, that is possible for a whole stack for real systems. And what we've done here, we have approximate, I mentioned this so far before, this interface with an homogenization step into a complex one. That means, for instance, we have approximate this in detail on the pore scale. Then we averaged over this and get then parameters that describes this highly complex pore scale driven processes on the REV, on the average scale. That's the idea. So that means end pore size classes with mean radius, evaluation drop formation conditions for each part. That means for each hole here. So then compute drop converged areas, liquid filled pores, then interface domain drop height, low dimension domain. And then what we have done with Karina together, she is a junior professor in she's a assistant professor in our team. She's from Norway. We have developed an homogenization concept for this drops for each time step. These are, for instance, the relative permeability saturation relationships. And now I come to the end. I will show you only few results to give you an impression. This is now the gas distributor here. This is the diffusive layer, and here is the reaction layer. Okay. Now we have the flow field here. We have the growth of the drops related to this reaction process, so that we have a continuous flux of gas from here to here, and related to this reaction, we get then an increase of water inside the system. That means we get the formation of the drops at the interface. So, and we have analyzed this. That means we have analyzed really this interface as an average quantity. That means simulation results, variations, several pore sizes. That means at first we have analyzed different pore sizes, and this is the drop volume over time. You see the drops, they will form, each drop will form. Then of course, we get the critical point where we get this detachment. The whole drops will be detached again and again, okay? But in this case, we have used everywhere the same pore size. That's the reason why we get this very smooth information, okay? If we use now different kind of cues, that means different kind of reaction rates, different kind of production of water in the system, then you see this has a strong influence on the detachment at the interface. You see here, if you have a low one, then you get this kind of detachment rate. If you have a very high one, then you get the difference, okay? The other one. Or this is here the drop volume of varying free flow velocity. Now we have different kind of velocities. That means we have in the gas distributor 3.5 to 4.5 meters. And you see directly, if you're higher velocity, if the velocity is higher in the gas distributor, then you get an earlier detachment of the drops itself. So, and these are the simulation in total. Let me come to the end. All these kind of things are implemented in our open source simulator Dumux. If you are interested on this, please have a look on our website. All these things are open source, and a lot of international partners are involved in this. And Bernd, my colleague and friend Bernd, is the head of this team. And you can find different kind of applications. A very funny one is related to the optimization of espresso. That's in cooperation with a company from Milano together. Okay. Let me come to my last slide. How simple do we want to choose our model? That's really the question for this kind of complexity that we have in mind to analyze. Finding the right level of model complexity involves analysis and multi-criteria. At first, we need a very, very good process and understanding. We need an understanding about the detailed description of the processes of flow and transport and deformation if necessary. Then the data must be available. That means we must have a good link to the data, how we can integrate this. I hope I've presented you this a little bit with this brain stuff. And last but not least, we need the relevant time and computation resources. And now I come to a sentence from Einstein. You know, Einstein is born not so far from Stuttgart in Ulm. It's a part of this state of Baden-Württemberg. And you know this sentence from Einstein: the model should be as simple as possible, but not simpler. And that is the key problem that we must solve. Mm-hmm. Last part, 2 slides. If you are interested to work with us together, we have a collaborative research center for this kind of topics in Stuttgart, entitled Interface-Driven Multi-Field Process and Porous Media Flow Transport and Deformation. And if you are interested on more information about this, if you would like to work with us together, if you would like to visit us, please let us know. You can find all these details on this website and on the social media too. Thank you much for listening to this, and I hope you will have a lot of questions. Thank you very much.